In this video, we will discuss about HiFi Cas9 system targeting KRAS mutation in vivo studies. So far, we have discussed about how they have developed their HiFi Cas9 system that can target only the mutated KRAS but not the wild type KRAS, and we have checked its efficiency in the in vitro studies also. In this video, the researchers has trying to use their HiFi Cas9 system by targeting KRAS mutations in the in vivo studies. So first they used the xenograft model where they took the adenovirus and transfect the cells with the adenovirus control and adenovirus HiFi Cas9 system, and then use this same and transduce the cells, and uh, they maintain the same MOI so that the transduction efficiencies remain same. MOI means multiplicity of infection or something similar to that. Yeah. So the idea is that they need to have the same transduction efficiencies so that they will get the results without any variability because of the difference in the transductions. And then they transduce the cells in different tumors uh, in the mouse. One is on the left side, one is on the right side. And after 60 days, they are able to see that the controlled adeno-associated virus tumors does not uh, gets any effect in the reductions in does not reduce its the tumor volume of the left hand side, which has been transduced by controlled adeno-associated virus, does not reduce, whereas tumor volume uh, got reduced for the tumors which are being transduced by adeno-associated virus with HiFi Cas9 system. The quantifications are shown in the panel B, where they have used two different cell lines. First one is H358 cell lines, and where they transduce the control in which they do not see any decrease, whereas when they use the HiFi Cas9 system they see a decrease. When they use the A427 cell lines, in this case also they see the similar results. When they use the HiFi Cas9 system they see reductions in the tumors. Then in the panel C, once they did their studies in the mouse tumors, they wanted to check its efficiencies on the PDX, it is patient derived xenograft. So in this case they took the surgical sections of the patients who are affected with the cancers and implant those cancers in the mice that can be seen here, and then treat these mice with different controls. Uh, the similar studies that they have done in the panel A, where on one side of the tumors they use the adeno-associated virus without HiFi Cas9, and in the other side they use the adeno-associated virus with HiFi Cas9, and then observe the tumor volume bi-weekly for 28 days. And what they are able to see is that in the case where they use KRAS G12C mutated xenograft, they are able to see that in the case of the control adeno-associated virus, they see a high the increase in the volume of the tumors is higher compared to the case where they use adeno-associated virus with HiFi Cas9. And the quantifications or the image of the tumors are also shown in the panel E, where we can see that the tumor volume when they use the HiFi Cas9 seems to be lower compared to the control for both the PDX1 or one xenograft and the second xenograft. And the results become quite evident when they did the quantification analysis, where we can see that even for these KRAS G12D mutations, when they use the adeno-associated virus without HiFi Cas9, the increase in the tumor volume is high compared to the case where they use adeno-associated virus with HiFi Cas9 system. So this further substantiate their previous findings that they observed from the in vitro studies, that the KRAS mutations, the HiFi KRAS mutations works well, or HiFi Cas9 system works well in the in vivo studies, and the results are similar to what they have seen in the in vitro studies. So this is what the researchers has shown in this figure. If you need to understand more about these results in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.